The overall goal of this procedure is to develop an in vitro model of smooth muscle contractile function that can be used to study the mechanisms of vascular dysfunctions, such as cerebral vasospasms, over disease-relevant timescales. This is accomplished with long-term vascular muscular thin films by first preparing elastomer substrates by serially spin-coating cover slips with a strip of PIPAM, followed by PDMS. The second step is to use a microfluidic deposition device to serially deposit genipin and fibronectin onto the surface in order to provide guidance cues for tissue self-organization on vascular muscular thin films. The final step is to perform a contractility assay by cutting vascular muscular thin films, inducing contraction, and relaxing contraction while imaging the sample. Ultimately, this approach yields tissues that maintain structural fidelity and functional contractility for up to 2 weeks in culture.
The main advantage of this technique over existing methods, like microcontact printed muscular thin films, is that tissue integrity and functional contractility are maintained for weeks, where tissues constructed using previous methods begin to degrade after 3 to 4 days in culture.
This method provides a platform for studying key questions in vascular disease, such as how the functional mechanics of arteries are altered during chronic disease progression.
Here, we are studying vascular mechanics, but this method can be applied to other model organ systems, such as organ-on-a-chip technologies for recapitulating cardiac or skeletal muscle function.
We first had the idea for this method when we aimed to study slow-developing vascular dysfunctions and found that current methods either could not be used to quantify vascular smooth muscle cell function or did not maintain integrity for a sufficient period of time.
Genipin microfluidic deposition provides a template for cell self-organization